Welcome to AI driven perception, learning and mapping for drones. Lecture 3, it talks about foundation in neural networks. Here we talk about single neuron model. What are the objectives of this particular lecture? Right? This is about foundation neural network module, single neuron model, how to represent a single neuron, what are the historical overview in single neuron models. We look at mathematical representation of this particular neuron model. Why do we need an activation function? We talk about different type of activation functions and how they represent the decision surfaces in that particular plane. And finally, we talk about simple Hebbian learning, and we take one example to understand how a single neuron model can learn a data, and we just go with simple working of some few samples and few steps to understand and appreciate. Okay. Now, let's look at a historical overview. In 1943, McCulloch introduced a single neuron model, neural network as a computing machine. Here the neural network perform logical computations as a theoretical foundation for AI. It's like AND gate, OR gate, NOT gate realization. In 1949, Donald Hebb introduced the concept of self-organized learning, a first model of synaptic plasticity and unsupervised learning. We are going to touch base on that and how we can use Hebbian learning for the single neuron model. We will discuss towards the end of this lecture. In 1958, Rosenblatt introduced Perceptron and supervised learning algorithm, the idea of a neuron that mimics the teacher. In 1960, Widrow and Hoff introduced the delta rule that talks about error minimization principles. It's a precursor to modern gradient descent algorithm. Okay. Now let's look at a biological neuron. Our single neuron comes from a single synapse, biological synapse, right? There is a source for a signal, that's what we call presynaptic neuron. And then you have a target, which is a postsynaptic neuron where the output goes to. And dendrites are the one which receives the signals from different neurons. Axon is the one which transmits the output to the other neurons. A synapse, right? Synapse is what you see a smaller region between the axons of the presynaptic neuron and dendrites of the postsynaptic neuron where it meets, right? That's a location called synapse. There are neurotransmitters in the synapse, which is basically a chemical messenger that neuron use to communicate across the synapse. It could be inhibitory or excitatory in nature, right? So basically what we are talking of, there is a presynaptic neuron which sends a signal to the postsynaptic neuron, and this is the synapse which we are trying to represent. These chemical messengers that what we are talking of, it can amplify the signal or it can suppress the signal, which indirectly represents the weights of the neuron. If the weights are positive and it amplifies, negative it could suppress. That's how we have to interpret this. Okay. So try to understand the signal flow through the neurons, right? We talked about it in that picture, but then I just want to give a more clarity here. The green one, right, is the dendrites which collects the electrical signals. You see these electrical signals are collected and they are coming to the nucleus, which is the cell body. The cell body integrates the incoming signal and generate signals, outgoing signal to the axon, right. Here is the nonlinear process take place, right. I receive all the signals, I perform certain process and produce an output, and that output flows through the axon. Axon passes the electrical signals to dendrites of another cells or to an effector cell, right. So if you look at it from a real world, we generate a data, the data are the features are coming in through dendrites and getting collected in the nucleus. Nucleus performs certain operations and the output of the operation pass through axon. Now let us try to represent this in a schematic of a single neuron. X1, X2, X3, Xm, M input signal coming from different presynaptic neurons. These are input signals coming through dendrites, and the synapse, right, has some chemical process, right, neurotransmitters that just sometimes inhibit or exhibit. We represent them as synaptic weights. Each neurons, each connection has a particular synaptic weight, and that is sent to cell body. Here it is like a synaptic potential has been computed, the W1 X1 plus W2 X2 plus Wm Xm, which is basically represent a summation j is equal to 1 to m Wj Xj. This is a synaptic potential, which is again in the cell body go through a nonlinear activation process, and the nonlinear process I am trying to represent as a function g, that is called activation function, and it produces an output y, right. Mathematically I can represent y is equal to g summation j is equal to 1 to m Wj Xj, right. What we saw, a single neuron without a bias. We will discuss about what is bias, why bias needed in the subsequent slides, but let us try to understand. X1, X2, Xm are the signal coming from different dendrites. X0 is the bias signal, right. It is passing through a synaptic weight called W0. So I am changing my synaptic potential going from j is equal to 0 to m Wj Xj, and it passed through activation function and an output. Mathematically, if I have to represent, y is function of W0 X0 plus the signal coming from all the dendrites Wij Xj. What is this W0? It is programmable bias term. Assuming that X0 is equal to 1, you can keep on changing the bias, right. You can update the bias in a way the network can handle the data variations. Okay. We will talk about it in the next slide. In a vector representation, I wanted to present y equal to g w transpose x. The dimension of w is m plus 1. The dimension of x is also m plus 1 cross 1. Okay. Now what is this bias term? Why the learnable bias terms are required, right? If you look at it, if you talk about input signal, all of them are 0, right. For a non-zero input, I want a non-zero output. Think about it, you are connecting this, the single neuron is connected to an actuation mechanism. There is no input and still I need something to hold my no load values, right. The motor need to be turned on. If the output is also 0, the motor is off, but 0 condition is like a base voltage that I had to provide. So I need a non-zero output, then I need a bias term, right. Think about it here, all xj's are 0, the term within the summation will become 0, what you get only x0 w0, right. This will provide a non-zero output. I need the bias term to get a non-zero output. This also helps the decision boundary. We will talk about the decision boundary in subsequent lectures. If I want to shift the boundary, see for example there are class 1 and class 2 and you want to separate them, right, by a boundary, and that is what we are trying to learn. We are trying to find the decision boundary which can segregate the data into separate classes. How do I segregate? How to distribute? If there is no bias, then the decision boundary always pass through origin, right. So we need bias such that the decision boundary can be shifted. If you look at it, for y should be greater than 0, then, and let us say x0 being x0 is equal to 1, the summation term should be less than w0, and with that we can find out our decision boundary. We will talk about it in a subsequent slide. You have to understand what is decision boundary, and that is when you will be able to appreciate. Most of the real world data is not always zero centered. Whatever you try to do based on the sample space, definitely the true distribution there will be some shift, and that shift if you want to handle, you need a bias term, right. Activation functions, they act like a threshold or a strength of an input signals, right. Having a bias will help you to shift the activation functions, right, that is also needed. If you look at a representation of it, shifting the activation function brings offset in the decision boundary, right. So, bias plays a very, very important role in learning as well as convergence. It is also control the orientation of the decision boundary, right, and the bias shift the decision boundary, your weights control the orientation of the decision boundary. So, you can shift your boundary and weights can shift the orientation of it, right. So, bias is very important for a neuron. Now, we talked about bias, we talked about the weights, but one important aspect is an activation function, right. We need this activation function to map the activation potential into nonlinear space. We can have a discrete activation, we can have a continuous activation functions. Let us talk about discrete activation function first. Discrete activation functions or hard limiting activation functions help in approximating the complex relationship. For example, unipolar, whenever x is greater than 0, or what I meant x here is activation potential is greater than 0, it is 1, otherwise 0. You can have a bipolar activation function, when activation potential is greater than 0 it is 1, otherwise it is minus 1, right. You have 1 and 0 or 1 and minus 1. You can have a ReLU function, right. Whenever x is greater than 0 you can call it x, but this does not come under discrete, but just for your understanding, right, otherwise it is 0. You can also have a piecewise linear activation function. It is a hard limiting function with a linear in certain range. For example, whenever the activation potential is greater than 1, it is 1, less than minus 1 it is minus 1, in between you have V, right. Between minus 1 to 1 it is linear, beyond 1 it is 1, beyond minus 1 it is minus 1. It is a hard limited, but between minus 1 to 1 it is linear. Okay. This helps very well in approximating complex mapping in the data. Now, let us understand the decision boundary, right. For decision boundary, let us assume activation function is operating in the linear range, right. If you look at the neuron model with a bipolar hard limit activation function, when I say bipolar hard limit activation function, when the potential is greater than 0 it is 1, otherwise it is minus 1, right. So, let us say there are two set of classes. There are sample points, right, you can see, which is in dark blue, and you have red. These are sample points, right, and the red ones belongs to class 2, the blue ones belongs to class 1, right, in a two-dimensional plane. What you see in the black line, which is the decision boundary, the boundary which helps in separating these two class set of samples, right. Now, the decision boundary which separates the m, I am showing it in two-dimensional. If I talk about an n-dimensional space, a boundary which separates the m-dimensional data into two categories, right. So, what we have here, you see the black line, that is my line separating these two classes, and that is a boundary we are talking of, which can be written as W1 x1 plus W2 x2 plus W0 x0 equal to 0. That is the decision boundary we are talking of, which helps in separating these two classes, right, in m-dimensional data or separating the line, right. So, what we are talking of, if you have a nonlinear activation function, a y which is g of x0 w0 plus summation j is equal to 1 to m wj xj. Now, the purpose of activation function, why do we need this activation function? The activation function is introduced in the model to have nonlinearity into the network, right. So, if you look at it, this is a linear activation function in the previous example. So, here I can have a simple line to separate, the data are very well separated, it is much easier to find the line. What if the data is like that? A set of samples, you see this red samples are in spiral, the dark blue samples are also in spiral. How do I separate? I cannot draw a simple line to separate. I may need to have a complex boundary like this to separate the data. So, introducing a nonlinearity helps this neuron to approximate nonlinear decision boundaries effectively. Okay. Let us look at different activation functions which are continuous in nature, which is often used in neural networks. They introduced to approximate the nonlinearity, they also helps in learning the complex relationship, right. Whenever we use this activation function, we use gradient computation to learn them. We will talk about it in subsequent lecture when I say what is gradient computations and all that. One example is unipolar activation function. Unipolar activation function, if you are to represent mathematically, y equal to 1 divided by 1 plus exponential minus x. X is nothing but activation potential, right. The range for this y is between 0 to 1. A typical activation of unipolar activation function is shown here, right. You see, if I use this activation function for the same problem, right. Now, how does my representation looks like? You can see that boundaries, right, of the single neuron model. What is the decision boundary? How does it project it into that space, right? Suppose if I use a bipolar activation function, bipolar activation function represent mathematically 2 divided by 1 plus e power minus x minus 1, right. X is nothing but activation potential, the range is minus 1 to 1. When I use a bipolar activation function, this is how my decision boundary looks like, right. So, a typical bipolar activation function I am just showing, this is nothing but tan hyperbolic function, okay. Whatever I am representing here is nothing but a tan hyperbolic function, right. I can have rectilinear unit, rectified linear unit, right. When x is greater than 0, it is x, otherwise 0. If you look at the range of this activation function, it goes 0 all the way to infinity. It is a typical ReLU activation function. You can see anything less than 0, it is 0, otherwise it is the same. If you try to use this activation function, I can nicely separate if the data is in first quadrant and fourth quadrant belongs to one class, and second and fourth quadrant belongs to one class, and first quadrant and third quadrant belongs to other class. I can approximate it very well, right. So, different activation function has its own impact at decision surfaces. We need to figure it out where it makes sense, right. How to use them, when to use them, we need to think about it, right. Now, we have to ask a question to ourselves. We have a single neuron, right, which receives input from dendrites x1 to xm, and it has a bias signal, and there is a synaptic weight. Can we learn this synaptic weight from the data, right? That is a question we need to answer. Mathematically, if we are representing, y is nothing but activation function times w transpose x, which is nothing but our synaptic potential, right. Now, can we learn them? Let us look at what Hebbian learning says, right. Donald Hebb's postulate, when the axon of a cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic changes take place in one or both cells such that as efficiency as one of the cell firing B is increased. Right? What does it mean? If both gets excited repeatedly and often, the synaptic weight started growing, these two started firing very often. Or this makes these neurons to fire together or getting wired together. Or we can say there is a positive correlation. If two neurons on this either side of the synapse activated simultaneously, then the synapse is strengthened further. Right? If there is a negative correlation, if they are activated asynchronously, the synapse is weakened further. Right? So, with this we can come up with a simple update rule. The change in weight, right, is a function of there is a symbol called eta. Eta is nothing but learning rate, whereas y is the output of the neuron and x is the input of the neuron. If x and y are similar, the weights get activated, strengthened further. If y and x are different, the weights is weakened further. Now, let us try to understand how to use Hebbian learning to find the weights of the neuron, right. Let us take a simple example. I am going to look at a 3-dimensional data, sorry, 3 samples with the 4-dimensional data, S1, S2, S3. These are 3 observations that are sampled from the distribution space, and each observation has a 4 dimensions, right. So, which means I have 4 inputs, 1, -2, 1.5, 0. This corresponds to sample 1. Sample 2, if you look at 1, -0.5, -2 and -1.5. Sample 3 is 0, 1, -1, 1.5. So, if you look at it, it is a 4-dimensional input. So, there is 4 dendrites which is giving signals to me, x1, x2, x3, x4. Corresponding synaptic weights are W1, W2, W3, W4, right. And I have a bias x0, W0. Summation j is equal to 0 to 4, Wj, Xj is my activation potential. I have selected a bipolar activation function here. When what is bipolar activation function? When the synaptic potential is greater than 0, it produces an output 1. When the synaptic potential is less than 0, it produces output -1. And that is my predicted output. If you notice here, the sample space does not have a teacher. There is no ground truth available to me. Okay. Now, what I am going to do? I am going to initialize these weights randomly. Now, let us make our life easier. Let us not have bias. I assume bias to be 0. Without bias, I am going to learn. I am assuming that these data are very well centered, right, and normalized. So, to make our life easier, I am going to assume x0, W0, x0 is 0. So, there is no effect of bias in the whole process. Okay. If I present the first sample, y is W transpose x into this activation function g, right. If I look at W transpose x, what do you get? 1 into 1 plus -1 into -2, 0 into 0.5 plus 0.5 into 0, which means what? You will get 1 plus 2, which is 3. 3 is greater than 0, so your output is 1, right. Now, what I am going to do? What is my update rule? The old weight plus eta times y into x. Eta is what we call learning rate. I am assuming eta to be 1 here to make our life easier, right, so that we can compute. So, old weight 1 into 1 is 1 plus the x, right. So, that becomes my new weight. Now, I can present the second sample. When I present the second sample, I am using the newer weight 2 -3 1.5 0.5, that is my new weight after learning. I am presenting my second sample. If you look at here, minus 3 into minus 0.5 is plus, which is 1.5, 2 plus 0.5 is 3.5, and 1.5 into minus 2, that becomes 3, and 0.5 into minus 1.5. So, the total term comes out to be negative 0.25. A signal is less than 0, so the output will become minus 1. In that case, the weight update, so old weight update plus eta is learning rate, which is 1, into y, which is minus 1, into x. So, that is my new weight. Now, let us look at the third sample. If you look at the third sample, this is updated weight after the two samples are presented, 1 minus 2.5, 3.5 and 2. W transpose x, right, the weight into x I am trying to do, and that activation potential is nothing but minus 3. My activation function is bipolar discrete activation. Because my activation potential is minus 3, which is less than 0, it is output is minus 1. So, weight update going to be old weight plus learning rate, which is 1, y, which is minus 1, into x, that is your new weight. We keep on repeating this process unless there is no change in synaptic weight, then we can call it. But if you look at this data, if you look at the output, you can see that the first sample is different from the sample 2 and 3. Sample 2 and 3 is giving negative weight, whereas the first sample is giving a positive weight, right. So, positive output. So, looks like first sample is different from second and third sample, but the only way to verify the observation right or not, you have to repeat the process till there is no change in weight is observed, then we can say the network has converged, and that we can use it to interpret how the samples are, right. This is a type of competitive learning. Thank you.